Welcome to the Honig Lab PrePPI website. In this video, we'll be discussing the protein networks and downloads panel on the website's results page. We'll be using the gene AP1G1, which corresponds to the UniProt ID 043747 as our example case. Navigating using the tab menu, we can arrive at the networks panel, which visualizes the predicted protein interaction network for the selected UniProt ID. Each node in the graph represents a protein, and clusters of tightly connected nodes often reflect proteins that participate in the same complex or biological pathway. Above the network, the legend explains the edge colors and styles. Dark gray lines indicate interactions with experimental support and databases, while light blue lines indicate interactions with no database evidence, highlighting potential novel predictions. Moreover, a solid line represents a structure-based or SM prediction, and a dashed line represents a peptide-based SLIM prediction. Users can customize how the network is displayed using the Network Layout drop-down menu. Layout options include circle, concentric, random, breadth first, and COSC. After selecting a layout, clicking Apply immediately redraws the network in the chosen style, which can make clusters and dense regions easier to visually separate. The network is also fully interactive. Users can click and drag nodes to reposition them, and can zoom in and out using the mouse scroll wheel or trackpad gestures. This makes it easier to focus on specific subregions of the cluster and inspect individual interactions. Below the network, a protein information panel updates dynamically when users hover over a node. This panel displays the protein's UniProt ID, protein name, gene name, organism, and a brief functional description. A View Results button is also provided, allowing users to open the detailed prediction results for that specific protein. On the right side of the network section is the top annotations for Interaction Network panel. This summarizes the most enriched functional annotation across the proteins in the network, reporting the annotation source, the term description, the number of proteins with the same annotation, the fold enrichment, the adjusted p-value, and the associated genes contributing to the enrichment. The full annotation results can be saved using the Download Annotations button, and a network image can be exported using Download PNG. Finally, the Downloads panel lets you quickly save results for the protein you're viewing. Use the Disorder panel to download residue-level disorder predictions, interfacial residues to export predicted interfacial residues, interactions to save the full interaction results table, and networks to export the interaction network output for offline use.